Okay, so Justin comes to us from most immediately from Maryland. Before postdocs in Maryland, he did his PhD at Iowa State on pigs and what he's going to talk about today. And then before that, he did his undergraduate at the University of Massachusetts in Amherst. So we're grateful. Justin now has a USDA postdoctoral fellow, and we're grateful that he's here in Lawrence.
Thank you both for the introduction. Yeah, so I'm going to tell you a bit about the postdoc work I did at the University of Maryland. This is going to be quite different from the previous work that I did as a PhD student, but also going to be quite a bit different from the work that I'm going to be doing here under my NIFA fellowship. But I think that this is of interest too, of course. So essentially, okay, now why is that? All right. Zoom wonders here. Okay. All right. So a chief ecological problem that I think every species has to overcome in order to really move forward evolutionarily, of course, is to find a way of reproducing successfully, right? So, I mean, these are like a very classical, basic biology aspect, right? And we know that there are a lot of different organisms out there, and there are a lot of different ways that organisms have come up with reproducing, right? And along with that, the ecological aspect of this has made it so that there are a lot of different ways that competing conspecifics try to gain access to a reproductive event, right? So I would say like one of the chief things that I am interested in as an evolutionary ecologist is how does this work and how does this maintain species interactions over evolutionary time, right? This is developed into a lot of different ways of dealing with this, right? So there's a lot of, especially when there's competitive events, there's a lot of pre and post copulatory sexual selection events that have come up, right? And the thing is that some organisms are better for understanding these than others, right? So
I'm just gonna move this so you can be near where you want to be.
Oh, okay.
Keep going.
All right. And especially in terms of the post-copulatory sexual selection events, such as sperm competition, which is what I'm going to talk about today, the basic biology of a lot of organisms make it very difficult to study this in vivo. And in particular, it kind of limits our ability to really understand what's happening. And that's where I'm going to try to, okay. That's where I'm going to try to say a better organism for studying a lot of aspects of post-copulatory sexual selection, but also reproduction in general, come from nematodes, right? And in particular, Caenorhabditis nematodes. So many people may be familiar with the exemplar taxon C. elegans. This has been, of course, the first organism that had its genome fully sequenced and published. But there are also a myriad of other reasons why nematodes are good things to use to study reproduction and reproductive events. Part of this, of course, is that they're small, they're easily manipulatable, they mate like gangbusters, right? A single individual will produce 500 offspring in her lifetime, and they do that over 3 or 4 days, right? You can freeze them, you can do almost anything you really want to with them. Of course, because of the genetic toolkit that they have, we have a lot of things that we can do. There are a lot of different isolates and mutated lines that you can use to really understand different aspects of their biology. For practical reasons, like I'm interested in, they're also transparent, as you can see, right? They have this really cool assembly line style gonad, right? Where basically immature oocytes push forward into fertilize embryos and then out of the animal, right? So you can systematically see the age of individual parts of the reproductive cycle within an individual, right? And as such, there's been a lot of work that's been done on understanding what's going on with Caenorhabditis nematodes in a reproductive context and in a myriad of other contexts, right? What I think is even more interesting, though, is that C. elegans is not the only nematode that's in the genus, right? There are dozens of nematode species that have been formally classified within Caenorhabditis as a genus. C. elegans, of course, is only one of them, right? This gives us a lot of power to kind of investigate why different ecological factors may contribute to the evolutionary history of individual species, but also of the group as a whole. Even more interesting, though, is that there is a variation in reproductive mode that's evolved independently multiple times within the genus, right? So like most nematodes, Caenorhabditis hails from a male-female obligately outcrossing ancestor, but 3 times at least within the genus, there's been the independent evolution of self-fertility through hermaphroditism, right? So these are like elegans, tropicalis, and briggsii, all produce oocytes as well as sperm within the same individual, right? So to quote, or at least to speak to my first postdoc advisor, Eric Higg, within his lab, he's interested in 2 fundamental questions. The first of which is, how does that happen, right? So how do you take a female body plan and produce sperm with it that produces viable offspring? And then where I kind of step in is, what are the ecological and evolutionary consequences of that change happening, right? And we have a lot of ability to really draw out what's happening here because we know so much about the evolutionary history of the group, right? And we can use these really close distinctions between selfing and obligately outcrossing species to make those comparisons and to do that kind of work. Even more interesting, and another aspect of weird nematode biology that I feel like is important to say here, is that even in species that have evolved self-fertility through hermaphroditism, you still can make males. And this is because unlike us, and unlike a lot of animals that have XX, XY, ZW sort of chromosomal systems, most nematodes have an XXXO chromosomal system, right? So females or hermaphrodites have 2 X's as their sex chromosomes, whereas males only have 1 copy of the X chromosome, right? This means that in hermaphroditic species, you'll have the independent arising of males through meiotic non-disjunction, and this happens naturally, right? But this also means that we can manipulate this adaptively for our research, right? So we can take a male once we see one, and we can artificially bottleneck again and again and again, so you have a lot of males to work with, right? And this is because when hermaphrodites self, they produce almost 100% XX progeny, but if you throw a male in the mix, then there's more of that XO that comes into the mix, right? And this will have important consequences for within patch sex ratios. And this is a really, really important thing to study, and it's not super trivial because there are pretty profound differences between the biology of selfers and of outcrossers, right? So what I'm going to show you here is the crossing of a Caenorhabditis male. So this is an obligately outcrossing male that we're mating with a C. briggsii hermaphrodite. So what we're going to see here, if I can get the video to play. So we'll see here in the outline, that's dotted line is the gonad arm of the hermaphrodite, right? So this is where you expect all the action for reproduction to be taking place. And in a second, we're going to be seeing little white dots. So those are sperm. Those are Caenorhabditis outcrossing sperm. If I can get this thing to go. So you'll see that the sperm have crawled, and this is another aspect, I guess, of nematode biology. I got to remember that there's not a lot of nematode people around here, but nematode sperm they're not flagellated. They have pseudopodia, so they crawl. So you'll see that they've crawled well beyond the confines of the gonad arm into the head, into the other body capsules, into the intestines, things of that nature. And this has pretty profound consequences for the animal, such that it results typically in sterilization or mortality, right? It's a reproductive isolating event, right? So this is a way that these 2 species have been driven apart, right? But it also highlights that there are chief differences between the sperm of outcrossing individuals and of selfing individuals. And more work has been done on that to show that selfing sperm is actually fundamentally different, right? It's lost a lot of the proteomic content that outcrossers have. It's also smaller in size. So when you have, let's say, a hermaphroditic or a selfing species male, they'll produce larger sperm that typically outcompete selfing sperm. However, they still get, I guess, lost over time. Right? So one of the important things, or one of the newly found items that has been found in outcrossing individuals or outcrossing species that's been lost within selfing species has been this gene male secreted short, or MSS. Right. So this is first identified in Caenorhabditis romanii, which is an outcrosser, and is now known to be a sperm surface protein that's essential for effective sperm competition in outcrossers that carry this. We know this because if you perform experiments where you knock the gene out in C. romanii, they produce sperm that is it's viable, so it'll move just like normal sperm, but it doesn't sire any animals, right? It doesn't compete as effectively as sperm that still have this MSS, right? We know a little bit more about it in terms of the fact that it's post-translationally glycosylated, and it also has a GPI anchor, which I'll get back to in a little bit. All of that is to say that we know that it has some sort of sticky membranous material on the surface of the sperm that likely has something to do with cell signaling, or at least with sort of interacting with the female tissues, which may give some sort of insight as to how it works, but we're not totally sure currently. Interestingly, though, MSS has been systematically lost in each of the selfing species that we know of within Caenorhabditis, right? So we know that it's an essential protein or an essential gene for the outcrossers, but we also know now that it's been lost within every species that does selfing. And this seems to make a lot of sense because these are, again, species where males are very rare, right? So it seems to make sense that this would be a thing that's lost. But it also provides us this opportunity of having these naive species that no longer have MSS, that we can reintroduce MSS to see how it works in vivo. And doing that shows that MSS is super competitive compared to male sperm that doesn't carry the MSS in these selfing species. So for the remainder of this talk, I'll be talking about C. briggsii, which is another one of the selfing species within Caenorhabditis. We don't need to get necessarily into the bombardment, but this is how it was introduced into C. briggsii. So if anyone has any questions about microparticle bombardment in the future, I'm more than happy to talk about it. It's a crazy method, right? And if you know anything about it's also used sometimes in plants, right? It's a transgenic method that predates CRISPR. It's also really, really useful for certain things in nematodes, particularly in places where you want to do mutations that work in the germline, right? In CRISPR, a lot of germline mutations get silenced, but in using microparticle bombardment here, it doesn't do that, right? All of that to say, I'll talk about that, and I'll cut to the chase and say it involves basically shotgunning gold particles into a bunch of embryos, and it kills a million worms, right? But some of them survive, and the ones that survive hopefully work, right? Anyway, when you do this, you put your gene of interest in, as well as a reporter gene. So in the case, the reporter gene that we have here is a green pharynx within the animal, so you can see that they picked it up, right? And doing that shows that the animal is super competitive when it carries MSS sperm as opposed to not carrying MSS sperm, right? So this is kind of a busy graph. All you really need to know is that this red here is MSS, right? So MSS carrying animals sire more progeny in mating experiments than wild type males or selfers, regardless of which mating order you put them in. So first or second, they still significantly produce more progeny right away than selfers. The presence of MSS also skews the population sex ratios for hermaphrodites. So this top line here is the proportion of males within a population over a number of generations, and the bottom line is when you just have wild type males contributing to the population. So as would be expected, over time, these males drop off if they are not carrying this sperm competitive protein. But MSS is so good at keeping males around and being a competitive protein against other males or against selfing sperm that it stays within the population a lot longer. And the proportion of males within the population stays around a lot longer. However, when you look at this over time, you see that within patch fecundity is higher for selfers when they don't have MSS. Right? So selfers over time produce significantly more progeny without the weight or the burden of males compared to not having it, right? So we have an understanding of MSS's competitive abilities, but we also understand why MSS has probably been lost in selfing species. Right? It's just simply over time, within patch fecundity is higher without it. And this is where I step in. When I started working in the lab, my chief question was, but how does MSS work? Right? So we know a lot about like what it does within populations. We know about why it was lost. We don't necessarily know anything about how it gains the competitive advantage that it has. And so I've come to the saddening realization that many of you haven't stared at nematode gonads as much as I have in the past, and shame on all of you for that. But so I'm going to walk you through the sort of action of a male nematode mating with a female or a hermaphroditic nematode. Over here on the far left is the vulva, right? So this is the opening to the outside for female or hermaphroditic nematodes. She'll have two gonad arms. They extend out in either direction. This is simply one of the two gonads, right? So here is the uterus. These are all fertilized embryos. You can see that they've already gone through a number of cell divisions, right? So they're going to be deposited outside of the animal, basically as things move this direction, right? They just get pushed out, right? So a male will come up, will interact with the female here, stick its spicule within the vulva, and then the sperm migrate, again crawl up this direction towards this organ here, which is the spermatheca, right? So in outcrossing sperm, the spermatheca, or sorry, outcrossing nematodes, the spermatheca is the sperm storage organ, right? So the sperm will ultimately arrive here, where it interacts with developing oocytes that are being pushed into the spermatheca. Right? In selfing nematodes, this is simply the sperm is produced elsewhere and then arrives here. Right? It doesn't come through the entire animal. Right? So the big point here is that the spermatheca is really where the action takes place. It's a site of exchange. Yeah.
Justin, can you just show that to the Zoom people?
Yeah.
With your arrow.
Yeah, sure. So here's the vulva. This is the uterus. Here's the spermatheca here, and then these are the developing oocytes, right? So these are immature. These are incapable of being fertilized currently. And yeah, the real action is where the spermatheca is, right? And that's where the sperm ultimately arrive, right? So in thinking about how MSS gains its competitive advantage, there are a couple of hypotheses that we can test, right? One of which is that MSS-laden sperm gain preferential access to the spermatheca through some sort of enhanced mobility, right? So they simply arrive at the spermatheca faster than MSS minus sperm, or sperm that doesn't carry the MSS, right? And if that were the case, one might expect to see something that looks like this, right? Where our red stars here indicate MSS positive sperm, the green stars indicate our MSS negative sperm. So one might expect to see a spatial segregation of MSS positive sperm in relation to the spermatheca compared to the MSS minus sperm. Alternatively, that might not exist. Instead, there might be some sort of fertilization advantage that the MSS positive sperm have that MSS minus sperm do not have, right? And if that were the case, you would not necessarily expect to see the spatial stratification, right? So you may see a more random spread of the sperm throughout the gonad arm each time. However, you would necessarily still see that MSS plus sperm sire more progeny than MSS minus sperm. So in order to tackle this, I should have gotten rid of this. This is for worm folk or for C. elegans folk. Basically, what we did was we used a feminized line of C. briggsii hermaphrodites. So these are C. briggsii hermaphrodites that no longer produce their own sperm, right? So that we don't have to complicate it with self-sperm, right? So we have virgins that we took. We'll effectively call these females. Virgin females, unmated. And we subjugated them to various mating combinations of MSS minus and MSS plus males that were stained with mitochondrial dyes, right? So the way that we did this is we either bathed them or fed them in these mitochondrial dyes, and they produce sperm that are fluorescent, right? And these fluorescences that you'll see are either red or green. Apologies to anyone in this room that's red-green colorblind. I got yelled at a conference once for this, and it's the way that we did it, unfortunately. But yeah, so then we mated them reciprocally for 2 to 4 hours, right? So MSS minus sperm first, then MSS plus sperm, vice versa, all the different iterative treatments you can think of. And then we imaged and quantified sperm localization using standard and confocal microscopy. I'll show you a bit about how that looks. And so when I say I quantified them based on these different parts or the localization, I put these up into different sections of the gonad arm. So I counted the number of sperm within each discrete unit of the gonad arms, including the vulva here, or the uteri. So the two uteruses, are there sperm there, or the spermathecae. And this is typically what we see, right? So you can't see them here, the people that are in the room, but the green here is MSS minus, red is MSS plus, right? So you'll see that there's a lot of mixture at the site of the spermathecae, which are here and here, right? So these are, this one's actually a mature oocyte that's moved through. It's probably already been fertilized, but it's being pushed into the uterus from the spermatheca, right? So you see this crowding of the sperm at the spermathecal valve, right? So they're vying for access to that oocyte as it comes in, right? But the point that I want to make here is that you see this sort of standard mixture, right? This is very common to what we see. And just for the point, so for simplicity's sake, for the rest of the talk, I'll show you some images that look like this, in which MSS plus is red. But I just want to mention that it works the other way too. It's not a dye effect, right? So you see the same sort of effects if and when you make MSS green as opposed to red. But for the rest of the talk, MSS is going to be red in these cases. So I'm going to try to do this now. So I apologize for this in advance because I've never figured out a good way of moving this. Can you guys still see this in Zoom land? All right. Yeah, so this is a proprietary Leica software thing that they I think intentionally butcher, and maybe someone out there will know better than me, but they seem to intentionally butcher the quality of the video so that you pay for a better package. This is the freeware. So instead, I'm just going to use this time to show you this video that I have on their freeware version of the package. Yeah. So what I want to show you here is basically what we see, right? So I'm also showing this because this was a huge pain to figure out how to do, and I'm really proud of myself for figuring out how to do it, right? So this is a 4D video, right? So this is taken on a confocal microscope. I think there are 5 to 8 Z planes present here over, I believe, 30 minutes of time, right? So the point here was to really understand, you can learn a lot about the positioning of the sperm at a discrete time unit when you take that picture, but you can learn a lot more by taking these videos, right? And you can understand what's going on a lot better. So I'm going to show you a max projection here. And this is going to be, again, the mating of a single individual. I'll play it a few times. I'll move this up to 2. All right, so basically what you're going to see here on the top, here is the spermatheca. You'll see a oocyte pushing into the spermatheca here. So this is the valve, the end of the valve. And then you'll see these sperm, again, green is MSS minus, red is MSS plus. And you'll basically see how they interact with that individual oocyte. So I'll play that again. So it gets pushed in, they swarm around it, and they try to get to the end of the spermathecal valve a second time. So again, this is really common, and it highlights the point that I wanted to show before, in which there doesn't seem to be a sort of mobility advantage given to MSS plus sperm, right? In this case, I believe the MSS minus was mated second, which is why there are more of that sperm there, because the MSS plus sperm already fertilized, right? But you can see that they both are seemingly capable of reaching the end of the spermathecal valve with each other, right? So one is not clearly faster than the other one. So I'll come back to this. If I can kicking myself in the butt for. Okay. Okay. Are you seeing presenter mode again? Okay, good. You're seeing presenter mode again? Oh, Jesus. I think I had to mirror these, duplicate. There we go. Now you're seeing, okay. All right. Although now we're seeing this here. Jeez. Really not gonna like having to do that again. All right. So yeah, so essentially we did this a lot, right? We were able to do this with a significant number of animals, right? And again, this number here is not necessarily super informative to you, but it was a lot of work for me to do, right? Turns out that when C. briggsii males inseminate females, they leave a copulatory plug, and that copulatory plug works, right? So getting this number was months and months worth of work, and now we're going to blast through it in 20 seconds on the slide. But we found no difference in the sperm count in the whole animal or any component of the gonad arm, right, within that animal. We also saw no evidence for spatial segregation, right? So it seems that the hypothesis that MSS plus sperm are capable of arriving at the spermatheca faster is not supported in this case. And instead, it might be supported that there's some sort of enhanced fertilization ability, which we're going to get into in just a second. So one of the things you might remember from the beginning of the presentation here, that when you mated in a gonad male with a briggsii female, you saw this invasive sperm behavior that killed the individual, or at least sterilized the individual, right? Nothing's really been known about what happens when you take a gonad sperm competition factor and put it into briggsii sperm, right? But one of the things that we started to find out was that they start to behave similarly, right? So here, this line indicates, again, the spermathecal valve, right? So on the right is the conventional gonad space, which is where you'd expect all the sperm to sit and be part of the normal routine of things happening, right? Anything beyond this place is ectopic, right? So these are developing oocytes that are not yet mature. Basically, there's no reason for a sperm to be back here, right? So it's interesting to see that simply putting MSS within these animals or within the sperm of these animals produces these invasive sperm. But even more interestingly, they don't behave like simple gonad sperm do, in that when we see these ectopic sperm, we only typically see one or two at a time. And they don't seem to have any sort of negative effect on fertilization or sterility or mortality or anything of that nature, right? They just seem to behave a little bit differently than MSS minus sperm. And again, we see this pretty frequently, okay? So here's a pretty clear picture of the spermathecal valve, of the spermatheca pushing out a fertilized embryo, right? On the other end, which is now sealed up, right? You see this MSS sperm sitting on top of the -1 oocyte, where it shouldn't be necessarily. And in fact, we saw that this was a very, very significant effect, right? So MSS minus sperm are significantly more likely to be found within the normal confines of the gonad. And MSS plus sperm are significantly more likely to be found in an ectopic location. So beyond the spermathecal valve where it should be. And this is actually present in 36% of the animals that we watched or that we saw, right? But again, negative effects due to this ectopic location were pretty minimal. So again, and this is going to be a fun thing again. It's very useful in this case, even more useful arguably than the previous case, to have videos of what this does, right? So what happens? What's the fate of an ectopic sperm? In order to do that, we managed to get a couple of videos. Again, this wasn't a super easy task. Yeah, if you've ever worked with confocal microscopy in the past, you'll know that the lasers are pretty intense. And I've blown up just as many worms as I've gotten actually to take useful images from. So again, these are challenging videos to take, but I think they're super rewarding. So what we're going to see here, this is just MSS sperm. So there's no MSS minus sperm here, but you're going to see an ovulation event happening in real time. So this fertilized embryo is going to be pushed into the uterus, and then you'll see the spermathecal valve close right here. But I want you to see sort of what happens when that valve closes. See this MSS sperm runs right in. So I'll make that a little faster. So it seems that as the ovulation takes place, there's some sort of attractant that the MSS plus sperm has that brings it in or sucks it into the spermathecal valve as it's closing up. I'll play it one more time. So now we have a mechanism of really about how the MSS sperm get ectopic, but what does it mean to be ectopic in this case? And so here is again another sperm competition video, right? So again, green is MSS minus, red is MSS plus. Show you. All right, so I want you to focus on this ectopic sperm that's here, right? So the spermathecal valve or the spermatheca is here, right? You have this pileup of sperm here, right? So I want you to see what happens to this ectopic sperm as the ovulation takes place. So it kind of walks around. I like to think it's trying to figure out what it's doing or what it's trying to do, right? I'll slow it down a little bit. Kind of goes back and forth. But then when the ovulation event takes place, it follows the oocyte into the spermatheca, right? And we don't necessarily have the power to say that that individual sperm fertilized that oocyte. It may have. We can't really see because unfortunately in this case, the spermathecal arm or the spermatheca went down, right? So we didn't have the Z plane to capture what happened there. But the point is, MSS plus sperm that get beyond the spermathecal valve are not necessarily doomed to stay there, right? They can follow it back, right? So it's not like it has this sort of brainless zombie style movement in either direction where it just ultimately kills. It may be able to follow back in. Which again indicates that there's just something fundamentally different about how MSS plus sperm work compared to MSS minus sperm. So now I'm gonna turn the slide. And we need to, right, that's the last time I have to switch it up. So there. Okay. Right. So now I had mentioned before that MSS, of course, has been systematically lost within the animals that carry them, or at least within selfing animals. And I also mentioned that it's GPI anchored, right? And we're going to use GPI anchor because that's the real term for it, and I'm not going to try to pronounce that, right? Essentially, is some component of cell signaling, right? That it's attached to the membrane, right? However, MSS is just one of many sperm competition proteins that's known within Caenorhabditis, right? We'll call these other ones MSRPs or MSS-related proteins, right? Some of which are still endogenous to selfing species, right? So the question arises, what's so special about MSS that it's been lost, but other sperm competition proteins or sperm surface proteins have not been lost, right? So for the purposes of this component, we can break these up into two main classes, right? Some of which are GPI anchored, just like MSS, some of which are not GPI anchored, right? So we can see using these comparisons if the sperm competition protein or the sperm competitive advantage that MSS has is due to the GPI anchor or not, right? And so to do that, I use microparticle bombardment again to produce these lines, one of which that we're calling MSRP8 is not GPI anchored, and one of which that is GPI anchored. Right? So then we can do all kinds of weird mating experiments, kind of like we've done in the past, but a little bit different, right? So I studied this in a number of different ways, and I'll say just briefly for the sake of time, they all resulted in the same answer that I'm going to show you in just a second, right? So we looked at this in 3 or 4 independent ways, and they all kind of lean towards what I'm about to show you. The easiest way, I guess, to describe this is basically doing these parentage assays. So essentially, we can mate MSS or MSRP3 or MSRP8 animals with a wild type hermaphrodite in this case, and we can see if they sire significantly more progeny than each other, right? So like if it were expected that MSRP3 lines that are now overexpressing these sperm surface proteins, I guess I should have mentioned that with the bombardment lines, we had them overexpress these endogenous sperm proteins. I don't know if that wasn't clear before, so I'm sorry about that. But so essentially what we're trying to do is figure out, does overexpressing these sperm surface proteins that are endogenous to the animal produce the competitive advantages that we see due to MSS, right? And if that were the case, you'd expect that they sire significantly a similar amount of progeny as MSS, right? You can do even more complex mating experiments where you throw in a wild type male to see if they can outcompete wild type males within the same individual. Essentially what we do is we harness these different fluorescent technologies that we have, or at least these different fluorescent transporters or reporters rather, to count progeny, right? So we mate these animals with an unlabeled hermaphrodite, and then over the next 24 hours or so, we count the number of individual offspring that have either the reporter gene here, so the glowing pharynx, or have the histone, the M-cherry histone wild type, right? So you can positively deduce who sired what progeny in that way. We ran this quickly and found out that basically nothing outperforms MSS, right? So MSRP3, MSRP8, neither of which produce significantly more progeny than each other, but MSS does produce significantly more than either, right? So basically we're at a point where we can't necessarily say what is causing MSS's competitive advantages. However, we can say it's doing something different, right? It should be noted that further validation of this is still required, right? Like we've done qPCR analysis to try to confirm that these things are overexpressing the transcripts that we think that they are. And there's some indication that they are, but we're recently going to be sending this out for RNA-seq analysis to count the number of transcripts, right? Alternatively, we have an undergrad back in Maryland that is she an undergrad? She might be a new grad student now, but she's going to be doing a histone analysis to try to see where these transcripts are being expressed. Because if they're not being expressed in the sperm, we may be seeing this for other reasons, right? So it looks like currently MSS is still special. We just don't know why, but we still do need to do some safeguards to make sure that we're confident in these results. So to summarize, MSS does not spatially stratify at the scale of the whole gonad, right? So this hypothesis that it moves faster or better than MSS minus sperm is not super supported. But we did find that MSS plus transgene is sufficient to make the sperm invasive, at least mildly so, right? Which highlights some of the differences between MSS minus sperm and why it might be doing or it might be having the competitive advantages that it has, such that I guess I'm saying within the the email that I sent out the other day, that they're more aggressive, right? So they have these fertilization advantages because they get in there faster or better than, and maybe not faster, but they bind to the female tissues better than MSS minus sperm. It appears that MSS is truly special in its competitive abilities, at least compared to overexpressing endogenous sperm surface proteins. And we can say that currently right now, we can deduce that it's not due to the GPI anchor, but we still need to do a little bit more work with that. So future work in the lab are to try to further dig into what makes MSS different, right? And the way that we're proposing to do this, or at least Eric is proposing to do this currently, is by making these MSS-MSRP hybrid constructs, right? So essentially what he's thinking about doing is taking an MSS signal or an MSS gene, or sorry, an MSRP gene, swapping in the signal peptide from MSS, seeing if that makes a difference, seeing if you can use that to say it's due to the signal peptide, or taking out the C terminus, things of that nature, right? All of this stuff might as well be witchcraft to me. I have no idea how exactly he's planning on doing it, but he's a lot better at that than I am for sure. And also, I'm not going to be the one that's doing that because my current project is on something completely different, right? So I wanted this to be abrupt because it kind of shows how abrupt my transition away from Caenorhabditis sperm competition world was to my current work, right? Which is going back to some of the observations I made as an early postdoc, but also as a PhD student, studying the fig wasp mutualism, which you may or may not know is this sort of model system for understanding the evolution and the stability of mutualisms. there are something like 900 fig species worldwide across the tropics and the neotropics, or the subtropics rather. And it's this classic case where the pollinating wasp oviposits eggs within a fig and it also pollinates the fig, right? So it has this thing where, you know, the pollinator needs the fig and the fig needs the pollinator, classic mutualism. My work in this as a PhD student was focused on parasitic diplogastric nematodes, which are entomopathogenic nematodes of the pollinating wasp, right? So they're vectored around by the wasp. They do some crazy stuff that I don't have enough time to talk about here, but I would love to talk to you about in the future if you'd like. Essentially what I found and the core of the work that I'm going to be doing is that it appears that these nematodes have some role in mitigating a pathogenic fungus that's also vectored by the pollinating wasp, right? So it seems that in that way, the pollinating or the nematode is both a mild parasite of the wasp and of the community, but also a mutualist in the community, right? So I'm trying to figure out how that works. I'm trying to figure out how that works in the context of where I've seen it in the past, which in this case is a Mexican fig species, a Mexican wild fig species, but also transferring some of that knowledge to the agriculturally relevant fig species Ficus carica within California, but also for Ficus carica wild isolates from Europe, right? So I'm going to be using this project to do some field experiments, field observations, but also be trying to grind my teeth on some bioinformatics using phylogenomics and biogeography. So with that, I mean, of course, I just blasted through that in not enough time. If you want to talk about that, I'm more than willing to talk to you about it for sure. But in the meantime, I don't know how we're doing on time here, but I want to thank the members of Eric Haig's lab at the University of Maryland and all the people that have helped me with this project. Our funding sources at the NSF. You can't see it in here, but there's this weird place or this weird consortium called the Baltimore Worm Club and in Maryland that is a collection of different worm labs in the area. I want to thank Jim Bever's lab for hosting me. And I want to thank all of you for your attention, for both the folk in Zoom land and also the folk in real land here. You guys are real too, I guess. I shouldn't say that, but yeah, so I'll be happy to take any questions if you have it. But thank you.